Hello and welcome to this Revision Monkey video on the required practicals for AQA Combined Foundation. And this is for the 2022 exam. So they've given you a heads up about what required practicals are going to be assessed. And for this exam, it is microscopes, food tests, and photosynthesis. If you're not sure if this is the right video for you, then do check with your teacher. This is for the combined scientists who are studying the trilogy version of combined science, which means you do 6 exams at the end of your course, all of which are 1 hour and 15 minutes long. Do also keep an eye on the description for the video because I'll hopefully put some questions for you on these required practicals in there, and I'll also put a link in there to the content for this Biology Paper 1 exam. Microscopes required practical. With the microscopes required practical, you need to know how to prepare a slide and how to focus that image under a microscope at different magnifications. So first of all, in the microscope, you've got the bit that you look into, and that is called the eyepiece. That often has a number on it, for example, times by 10. We'll come back to that later. You've got the objective lenses here, and often they have different numbers on. For example, you might have times 4, 10 and 40, for example. Now, to work out what magnification you're using, you multiply the objective lens by the eyepiece number. For example, if you looked at 4, that would be 4 times 10, which would be 40 times magnified. The 10 lens would be 10 times 10, which would be 100 times magnified, and the 40 times 10, which is 400 times magnified. This here is the stage where you put your slide that you want to view, and often on the arm here you might have 1 or perhaps 2 focusing wheels. The larger one will be for the coarse focus, so you can just call that a coarse focus wheel, and the other one would be for smaller adjustments, which is the fine focus wheel. So now I'm going to take you through the steps to carry out this required practical. First of all, to prepare the slide, you need to spread the cells on the slide, add a dye, for example iodine, to stain the cells, and then add a cover slip on top, and this is just a small transparent square so that you don't get any of the dye onto your lenses. Place the slide on the stage and use the grips to keep it in place. Select first the lowest magnification objective lens to view your cells. So for example, in this case, we'd make sure we start off with 4 times 10, which is 40 times magnified. We use the coarse focus wheel and then the fine focus wheel to focus the image. And we would then draw the image using a pencil and no shading of any of the subcellular parts. So for example, if we're drawing an animal cell, we might draw the cell like so. With the nucleus, we wouldn't shade it in, but we'd make sure we labelled the bits that we saw, for example, the nucleus and the cell membrane. Finally then, we would then change the lens to a higher magnification and refocus the image. As an extension to this required practical, you might be asked to do a magnification calculation, for which you need to remember this equation. Magnification equals image size over real size. For example, a bacterial cell was viewed at 100 times magnification under a microscope. The width of the bacteria cell in the photograph, over here, is 20 millimeters. What is the real width of the bacterial cell? Give your answer in micrometers. So lots to do here. First of all, we need to work out what we've got. We've got a magnification, and we've got the size of a photograph, which is an image. So this is our image size, and we need to calculate our real size. So first we'd rearrange this equation. So it would be real equals image over magnification, and then we put our numbers in. The real size of the bacterial cell will equal 20 divided by 100. And this will give our answer in millimetres. So our answer is 0.2 millimetres. Now we need to change our answer to micrometres, and to do that we times by 1,000. So our final answer would be 200 micrometres. Food tests require practical. So there's 3 food tests you need to be aware of. In your revision guide they may talk about a test for fats as well using Sudan 3. However, because that's quite a nasty chemical, they've removed that as part of the required practical. So you shouldn't be asked on fats, you should just be asked on carbohydrates, which split into complex carbohydrates like starch and simple carbohydrates like sugar, and then protein. But first of all, you need to prepare your food sample, and you do this by crushing up your food in here using a pestle and mortar. And then you'd mix it with a little bit of water in a test tube or a boiling tube to make a food solution. So we could do 3 different tests. We could do a test for starch. So if we have our food solution in here. So the test for starch is to add iodine, and if iodine detects starch, there will be a colour change from orange to black. So if this food sample contains starch, it would turn black. So to test for sugar, we'd put some of our food solution inside a test tube, and we would add Benedict's solution. Benedict's solution is blue, but importantly we also have to put this in a water bath. So you could either heat it up and try and maintain the temperature, or you could use an electronic water bath, and it needs to be around 75 degrees C, so quite a hot water bath. And a positive result, if we put our Benedict's inside, heat it up in the water bath, a positive result would be a change from blue to orange. Now you may well see in your revision guide other colours as well, because if it's got a little bit of sugar in, it might go a green colour. If it's got a lot of sugar in, it might go orange, or maybe even a brick red colour. Okay, so in the exam they might talk about green, orange, or brick red. But if in doubt, remember blue to orange. So to test for protein, our final food test, we would add a food sample into our test tube. And the test for protein is to add Biuret solution, and that's quite a light blue solution that we add in, and that will turn to purple. Okay, it's quite a light purple, that's why I put light purple, but you still get the mark if you put purple in there as well. So if protein's present, you will get the colour change from blue to purple. Measuring the rate of photosynthesis required practical. In this practical, we submerge some pondweed in water, and the number of bubbles it gives off per minute gives us a rate of photosynthesis, and the investigation looks normally at how light intensity affects the rate of photosynthesis by moving a lamp closer or further away. For example, if you had your lamp far away pointing at the pondweed, you might get a few bubbles coming off in that minute. If you then moved it closer, you might get a few more bubbles given off in that minute. If you moved it closer again, increasing the light intensity that's reaching the pondweed, in that minute you may well get a few more bubbles giving off, indicating a higher rate of photosynthesis as you move the lamp closer to the pondweed. So let me run through the main steps of the practical that you need to get down if asked to write a method. Cut 10 centimetres of pondweed and submerge this in water in a boiling tube. Clamp the boiling tube to a clamp stand. I haven't shown this here, but you may well have set up a clamp stand just to make sure that stays in the right position, clamping it there like so. Measure 50 centimetres away from the clamp stand using a ruler and place a lamp here directed at the pondweed. Turn on a stopwatch, record the number of bubbles produced by the pondweed in 1 minute. Repeat this experiment for the following distances: 40 centimetres, make sure you list them, 30 centimetres, 20 centimetres, 10 centimetres, each time recording the number of bubbles produced by the pondweed in 1 minute. So our variables are important here. Our independent variable is the distance between the lamp and the pondweed. The dependent variable is the number of bubbles per minute. And important control variables, things like type of pondweed, the length of pondweed, the power of the lamp you're using. For example, you might want to say you're going to keep it at a 40-watt bulb or something like that, and the temperature. Now, with regards to temperature, you may well see a transparent heat shield in front of the lamp, which will stop the lamp from heating up the water. Or you may well see that the investigation makes sure it uses an LED lamp, a light emitting diode, rather than the filament lamp, and that will reduce the heat given off and therefore help you control the temperature in the experiment.